Hello, people. You better learn. Okay, we're going to go over frequency of cell division. There's going to be a lot of noise all the time. I apologize in advance. So frequency of cell division. Basically, the amount of time a cell will divide varies on the cell type. Essentially, an embryo will go through the cell cycle, which is right here. Remember, it's on my floor. Every 20 minutes, which would make sense because an embryo is going to be dividing very frequently. Skin cells are another cell that divides really frequently. You're looking at about an entire new, one skin cell will divide between 12 to 24 hours. It'll go through this cycle. Liver cells, remember they divide quite often. About every 2 years, your entire liver is completely new. So these cells have a very fast rate of cell division. Nerve cells and muscle cells are two cells that will not divide after maturity. They will stay at the G0 phase. And the G0 phase is the area right before, after, kind of right in this area right here, right here. And basically, yeah, oh, okay, Harperly, I'm doing this. Okay, good. Okay, so the G0 area would be right in here. And basically, this is what a cell does when it's not dividing, and it's actually doing whatever the cell is supposed to be doing. So it would make sense that nerve and muscle cells are never in the cell cycle because you can't have your nerve cell decide it's going to take a break. So here is an overview of the cell cycle controls. Basically, you're going to need to know this for your test. You have two ways that the cell cycle can be controlled, internally and externally. Internally, you have things called cyclin and cyclin-dependent kinases. These guys will basically determine if the cell gets to go to the next checkpoint. So when we say internal, we're talking about what's actually happening inside the cell cycle. Externally, you have growth factors. We're going to go into that a little later. And a thing where it basically can check the density of the neighbor cells. This is a picture showing the cyclin and the cyclin-dependent kinases. Your book does a very good job of describing this. Cancer cells usually don't exhibit either of these. All right, so here are our checkpoints. These are the things that get to decide if a cell keeps going in the cell cycle. Remember, cancer is the inability to control this cell cycle. Basically, cells just keep going. Oops, sorry about that. You have 3 checkpoints, and we did this last year. We talked about it, and we even did the cell cycle game. But this is a checkpoint, G1. And then, so it's right before G1. Basically, the cell will do a checkpoint to say, okay, is the cell big enough? Does it have all the chromosome it needs? If it doesn't meet that criteria, then it undergoes apoptosis. That's cell death. G2 is another checkpoint. It occurs right after the second gap phase. And then the third and final checkpoint in the cell cycle is it's in the middle of mitosis, and we'll talk about that a little later. All right, so, I guess this was on the next one. G1 to the S phase, so that's right here. This phase right here is the most critical. This is can DNA synthesis begin? If it's a no-go, And that meaning that the cell, we don't need more cells, it'll go and sit in G naught and it'll stop dividing. G2 is the G2 to mitosis is the second checkpoint. What they're checking for here is, has the DNA synthesis been completed correctly? And after G2, that checkpoint is the commitment whether or not the cell is going to go into mitosis. If it gets the yes, then it goes straight through mitosis. The spindle checkpoint that occurs in mitosis is basically the check to say, are all chromosomes attached to the spindle? Can the sister chromatids attach separate or correctly? Remember, the point of the spindle attaching to those kinetochores that are on the centromere is for those sister chromatids to get ripped apart. This checkpoint, if there is an issue and the spindle is not attached to that kinetochore, you're going to have chromosome separation errors. That's where we get the sticky chromosome that happens with Down syndrome. Okay. If the cell receives the go-ahead at G1, then the cell's going to continue on into the cell cycle. If it doesn't, and I alluded to this in the last slide, it'll go into G naught. And don't think G naught's just a useless stage. That's where the cell does what it should be doing. If it's a liver cell, it's detoxing. If it's a, lung cell, it's going to be doing whatever lung cells do. So again, here's our G naught phase. It's in the non-dividing stage, and most human cells are in the G naught phase. Liver cells will go into the G naught, but then they're called back to the cell cycle. And again, that's where we have these external and internal cues that will tell the cell we need a new cell, or we need to reproduce. Nerve and muscle cells, these are highly specialized. They stay in G naught and they can never divide. This is where we have the kind of much needed research for stem cell. Because if we could do stem cell research and find out ways to put stem cells in and regrow nerve cells or muscle cells, then we could heal injuries that we normally could not. Muscle cell injuries, some of you guys might have. If you've ever looked and you notice there's a divot in your muscle, I have it a lot of times. I've had one horse that has that, where there's actually a divot in the muscle tissue. And then some people are going to be saying, yeah, but I get big muscles. Isn't that my muscles growing? Your muscle cells themselves can get larger, but you cannot increase the number of the muscle cells. The cells can grow, but you don't get more. All right. So how do cells know when to divide? This is the internal and external signals. Okay. Harper, could you be a little quieter? Thank you. All right. Sorry about that. It's going to be a lot of these. So how do cells know when to divide? We have internal and external signals. This is an example here where they did an experiment where a cell was undergoing mitosis. They connected it to a cell that was in the G1 phase. When they put them together, basically mitosis was activated in the cell that was in the G1 phase. It shouldn't have been activated, but that's a hint. That means that there was some kind of protein telling the cell to start mitosis here. And when this cell was connected to the G1 cell, suddenly this guy started to divide. Okay? We think those signals are usually proteins. So again, the cell cycle controls, and I'm going to finish this video and then I'll show you a little video of a demo. But we have basically, these are going to be your internal controls. And you must understand this for your test. You have two main deals. You have cyclins and you have protein kinases, also called CDKs. Cyclins are going to be regulatory proteins of the cell cycle, and they will fluctuate in their number. Basically, these cyclins, the amount of cyclin, when that will go up and down, depending on what point in the cell cycle it's in. When the cyclin increases, it basically turns on this CDK. So what happens is the cyclin will bond with the CDKs, and that will activate this complex. When the cyclin and the CDKs are together, we call it a CDK cyclin complex. It's essentially the cyclins turn on the CDKs, okay? So that's what this picture is showing. When the cyclin goes into the CDK, it turns it on, all right? MPF, your book talks a lot about, that is just an example of a cyclin CDK complex, okay? The way it works with these guys, this is showing you, I'm going to wait for the next slide to explain that, but this is showing you where MPF, which is again a CDK, its activity increases when the cyclin associated with it peaks. Its activity decreases immediately when the cyclin number decreases immediately. So here you have the cyclin building up, building up, building up throughout G1, S, and G2. It hits mitosis, that is the highest amount of cyclin. And look at what happens with the CDK number. CDK has suddenly spiked its activity, and this CDK again is called MPF. It spiked its activity, and now it's at the highest activity level. And that is where this is in the mitosis checkpoint, okay? So it pushes it through. Once it gets done with that checkpoint, this number drops, the amount of MPF activity. And if you look, the cyclin number drops as well. So what we could tell and what we need to know, remember that's in red, when cyclin reaches the sufficient concentration, it activates that CDK molecule. In our example, MPF is basically turned on when the cyclin associated with it reaches this threshold level. Now, all these CDK deals, these molecules, will shut themselves off. So this is showing, this is MPF. It's an example of a CDK, okay? So here is MPF. Also, again, it is an example of a CDK. Is basically connects to the cyclin. It becomes activated. After it's reached the point of mitosis that it's pushed it through the checkpoint, this cyclin gets degraded. It is degraded by MPF. MPF will basically shut itself off by ripping apart its own cyclin. And then it's really cool because then the CDK is basically recycled back, and the cyclin concentration is built back up, and the whole process keeps going. MPF triggers the cell's passage past the G2 checkpoint into mitosis. That you just have to know that that's what MPF does. Again, I know I've said this a lot, but it is an example of a CDK, okay? But basically, it triggers this passage into mitosis. And this is just a very good picture. It's just showing you CDK and cyclin coming together. Cyclin is decreased at a specific point because, I've said this, but we're going to say it again, because the cell will, the CDK will initiate its own destruction. It basically rips apart its own cyclin. This is its own slide. It's in bold. Don't you think that's going to be a test question? Cyclin and cyclin-dependent kinases will basically drive from the 3 checkpoints from one phase of the cell cycle to the next. And this is kind of a cool thing. The genes for these CDK and cyclin have been highly conserved through evolution, and basically the same CDK and cyclin genes that we use in our cell cycle are also used for plants, insects, and yeast, which makes genetic engineering quite fabulous because we're able to basically use the same kind of tools to maybe have the yeast or bacteria produce our own genes. All right. Now, these are the external, example of two external signals and then an internal signal. The internal signal that influences cell division would be growth factors. Those are those proteins released by certain cells, and they stimulate other cells to divide. Density-dependent inhibition, which is an external thing, and anchorage dependence. Basically, density-dependent is if cells are crowded and touching each other, it stops them. Anchorage dependence means that if cells are attached to, they have to be attached to something, like a petri dish. This is showing you the difference. So they have to anchor, normal cells would have to anchor to the actual petri dish. And so they'll just form a single layer. Whereas other cells might, and if you took two of them off, then that would kind of inspire new cells to basically fill in this gap. But they're not going to go on top of each other. That is an example of density dependent and anchor dependence. Cancer cells, on the other hand, do not care that they're attached to the bottom of the petri dish, and they are not density dependent, and they will literally grow on top of each other. Okay? All right, I'm going to stop the audio now, and please watch the demo and then come back and we'll chat a little bit about cancer cells.